So at the end of S phase, we're going to have our DNA totally duplicated. And this is where you'll see the sister chromatid structure that we were looking at before. Now we'll move on to G2 phase, which is the second gap phase. And again, I really want you to remember that this is when the cell is living its normal life. It's doing normal cell stuff, but it's also growing and it is getting ready to divide. So all three of these normal parts of the cell's life are called interphase. So interphase is G1, S, and G2. All of this together is called interphase. And during this time, so most of the cell's life, about 90% of the cell's life, is not dividing. Okay, this is something that students often get confused about. Cells do other stuff besides divide. Mostly they're just living their normal life. But towards the end of their life, they go into another phase called M phase. And M phase has two parts. The mitosis or meiosis, depending on what you're looking at. The mitosis part and the cytokinesis part. Mitosis is basically all of the different steps of division and separating the chromosomes and everything. And cytokinesis is just the physical splitting of the cells. And together, these two parts make up M phase. So if you were asked, how many parts are there in the cell cycle? Well, you could say two. There's M phase and there's interphase. But interphase is really made of three smaller parts, G1, S, and G2. And M, in the mitotic phase, is actually made of two smaller parts, mitosis and cytokinesis. So what is mitosis? So during mitosis, we're separating that DNA and we're actually breaking the nucleus and making new nuclei, the plural for nucleus. How do the chromosomes actually move? How do they get moved around and separated? Well, that's done using something called mitotic spindles. And mitotic spindles are made of microtubules. They grab onto the chromosomes and move them around the cell. So the mitotic spindles are doing a lot of the work here. They're helping to keep everything organized and move the chromosomes where they need to go. By the end of mitosis, the two sister chromatids, which are copies of each other, will move to opposite parts of the cell, and they're going to be in the new nuclei that form. So let's look at some of the steps of mitosis. Well, first, what does a cell look like when it's not in mitosis? Remember, 90% of its life, it's in interphase. During interphase, if you look at a cell, you're going to see that it has a normal nucleus and the DNA will be in its loose chromatin form, so you won't really be able to see individual DNA molecules, but you will be able to see chromatin, which will just look like a bowl of spaghetti. Now, once we enter mitosis, we're going to have 5 different phases here. And some books and videos will say 4 phases. They kind of skip prometaphase. But in the Campbell curriculum, we're going to use this step in between prophase and metaphase, which is called prometaphase. So what I'm going to do now is just kind of go over the most important things that are happening in each step. So the first step is prophase. In prophase, the nuclear envelope, or the outside of the nucleus, is going to break down. It's going to start to break. And also our chromatin is going to condense and pack together very tightly into a structure called a chromosome, a condensed form of DNA. And you can see these sister chromatid are joined together at the centromere. And lastly, the mitotic spindles are going to start to grow out of the centrosomes. This is what's happening during prophase. During prometaphase, what's happening is the nuclear envelope is almost all the way gone, and the mitotic spindles are going to connect to something called the kinetochore, which is part of the chromosome. That's just the part that holds on to the chromosome. So it's the mitotic spindle that's where it grabs the chromosome, is the kinetochore.